Right now, inside your body, there is a microscopic bouncer physically dragging a rowdy, misfolded protein out of a club to be destroyed.
Yeah, and if that cellular bouncer fails, the result isn't just a ruined party. It is literally the biological origin of diseases like Alzheimer's.
Welcome to a deep dive into the microscopic foundation of human disease. Whether you are a medical student prepping for boards, a young healthcare professional, or just like a self-taught science enthusiast, we are custom tailoring this session to shortcut your way to being well-informed.
Today we are pulling from Lippincott's Pocket Pathology by Donna E. Hansel and Renee Z. Dences. Our mission is to basically give you a comprehensive, clear, and highly scientific summary of the key pathways of pathology from that text.
Right, so we are going to trace the entire life cycle of a disease, starting from how a single cell adapts to stress, what happens when it fails, how the tissue responds, and finally how that can cascade into systemic shock.
Exactly. Because before a patient ever feels a symptom, before a doctor can even make a diagnosis, the pathology always begins at the microscopic level. It starts when a cell's internal environment is threatened.
And the text outlines 5 main ways a cell can adapt to that stress. Because it doesn't just give up and die immediately, right?
No, not at all. Cells are highly dynamic. When a stressor hits, the first adaptive response we usually see is atrophy. This is a deliberate decrease in cell size and function.
So it just shrinks.
Right. If there is a reduced functional demand, like say a limb is immobilized in a cast, or if there's a chronic lack of blood flow, which is ischemia, the cell shrinks to conserve resources. It powers down.
Okay, so the opposite of that would be hypertrophy, right? Where the cells physically increase in size.
Exactly. And the text gives a really great example of this with systemic hypertension. The heart has to pump harder against that high blood pressure, right?
Yeah.
So the individual cardiac muscle cells actually synthesize more structural proteins.
They literally bulk up to handle the mechanical load.
Yeah. But it's not just mechanical. If patient is taking certain drugs, like the barbiturate phenobarbital, the liver cells have to manage that chemical burden.
Oh, right. So they expand their smooth endoplasmic reticulum to detoxify the drug.
Exactly. The cell is building more internal factory floor space to handle the chemicals. Then the third adaptation is hyperplasia. This is different because the cells aren't getting bigger, they are multiplying. They increase in number.
Like how going to a high altitude causes your body to make more red blood cells.
Right. Or how estrogen increases uterine size at puberty. That's physiological. But it gets pathological when, for example, chronic friction from bad shoes causes skin cells to rapidly multiply and form a callus.
Okay, so atrophy, hypertrophy, hyperplasia. But the fourth one is where things get kind of crazy. Metaplasia.
Yeah, metaplasia is wild. It's a reversible change where one fully differentiated adult cell type is completely replaced by another cell type just to survive a harsh environment.
The classic example being a smoker's lungs, right?
Yes. Normal respiratory lining has delicate ciliated columnar cells that sweep out debris, but smoke is toxic. So the body replaces those delicate cells with tough, stratified, squamous epithelium.
But the trade-off is terrible because now you don't have those sweeping cilia anymore, so you're super vulnerable to infection.
Exactly. We see the same thing in Barrett's esophagus. A patient has chronic acid reflux, so the normal squamous cells in the lower esophagus are bathed in stomach acid.
Which they totally aren't built for.
Right. So they change into simple columnar cells with goblet cells, which is the exact acid-resistant cell type you would normally find in the stomach or intestines.
That is amazing biology, but it sets up the fifth response, which is dysplasia, where the growth becomes disordered and irregular.
Yeah, dysplasia is adaptation losing control. The cells vary in size, their nuclei get dark and enlarged, and the tissue loses its normal architecture. It often happens right inside those areas of metaplasia.
And that is super dangerous because dysplasia is basically precancerous, right?
It is heavily monitored because severe dysplasia is the immediate precursor to invasive carcinoma. It shares a lot of features with actual cancer.
Okay, so what if the cell can't adapt? Like the stress is too much or there's a genetic defect? The text says they become microscopic hoarders and start accumulating substances.
Yes, intracellular storage. In diabetes or chronic alcoholism, the liver cells can't process triglycerides, so fat builds up inside the cell. It literally pushes the nucleus to the side.
Wow. And then there's glycogen accumulation, like in McArdle disease?
Right, McArdle disease is a genetic mutation where patients lack muscle phosphorylase, so they physically cannot break down the glycogen stored in their muscles.
So it just piles up and causes terrible cramping when they exercise.
Exactly. And then, of course, cholesterol accumulation. Macrophages in the arterial walls try to clean up excess lipid, gorge themselves on cholesterol, and turn into what we call foam cells.
And those foam cells cause atherosclerosis. But I mean, the most fascinating part to me was the protein quality control, because proteins have to be folded into a very specific 3D shape to work.
Yeah, normal proteins get escorted by chaperone proteins, but if they misfold, they become highly toxic.
Which brings us back to the bouncer analogy from the intro. The cell has this ubiquitin proteasome machinery. It slaps a ubiquitin tag on the rowdy, misfolded protein.
And that tag tells the proteasome, which is the security team, to drag it away and destroy it. But when that fails, you get serious diseases like alpha-1 antitrypsin deficiency.
Right, where the liver makes this protein that's supposed to go protect the lungs, but it misfolds.
So it evades the bouncer, aggregates in the liver, and causes cirrhosis. And because it never reaches the lungs, normal enzymes just chew up the lung tissue, causing emphysema.
It's a dual organ failure from one tiny misfolded protein. And failing to clear proteins is also the hallmark of neurodegenerative diseases, right?
Absolutely. Prion diseases, Parkinson's where alpha-synuclein clumps into Lewy bodies, and Alzheimer's with amyloid beta tangles. It all comes back to failed quality control.
Okay, so if the bouncers fail or the stress is just too much, the cell is pushed past the point of no return. It dies.
Yes, cell injury and death. The quintessential mechanism here is ischemic injury, which is a sudden cutoff of blood flow.
So no blood means no oxygen.
And no oxygen means the mitochondria can't make ATP. The energy currency drops to zero.
Which means the sodium-potassium pump on the cell membrane completely shuts down. And since it can't pump sodium out, sodium rushes in and water follows it.
So the cell swells up like a balloon. But the terminal event is calcium. Calcium floods into the cytoplasm and acts like a biological trigger.
Right. It turns on these destructive enzymes called phospholipases, which literally tear the cell membrane apart from the inside.
Exactly. Once that membrane breaks, the cell is dead. This messy, uncontrolled death is called necrosis. All the internal enzymes spill out into the tissue.
And pathologists look for specific patterns of necrosis, right? Like coagulative necrosis.
Right. Coagulative necrosis is typical of an ischemic infarct, like a heart attack. The sudden drop in pH denatures the cell's proteins, including its own enzymes.
So it basically freezes the dead cell in place.
Yes, the tissue architecture is preserved for a few days. Under a microscope, they look intensely pink, or eosinophilic, like ghost cells.
But liquefactive necrosis is totally different. That's when the tissue just dissolves into liquid, like with a bacterial abscess or pus.
Exactly. Or a brain cyst after a stroke. Now, we contrast all of that messy necrosis with apoptosis, which is tidy, programmed cell death.
The cell basically shrinks, packages itself up nicely, and neighbor cells eat it without causing any inflammation.
Right, driven by specific cascades, like the BCL2 gene family.
Okay, wait. I have a specific question about the ischemic injury part. I read something in the text that confused me.
Oh, sure. What is it?
So, if cutting off the blood and dropping oxygen causes the ATP to fail and kills the cell, why does the text say that restoring blood flow, like reperfusion, can cause even more injury? Shouldn't fresh oxygen save it?
It seemed totally counterintuitive. But when the cell is severely damaged from ischemia, its mitochondria are physically broken. If you suddenly flood that broken system with oxygen, it can't process it normally.
Oh, so it misfires.
Exactly. It inappropriately reduces the oxygen and generates massive amounts of reactive oxygen species, or ROS, free radicals.
And free radicals are incredibly unstable, so they just crash around destroying proteins and DNA. So the fresh oxygen actually causes severe oxidative stress.
Exactly. Now, when necrosis happens and that messy spill occurs, it triggers the tissue's response. Inflammation.
The local cleanup crew. I like to think of acute inflammation as the fire department. They rush in, cause some fluid leakage, which is the swelling, and the neutrophils start aggressively eating up the dead debris.
But if they can't clear it, or the stress persists, it shifts to chronic inflammation. The neutrophils leave, and the macrophages and lymphocytes move in.
The demolition crew.
Right, and they aberrantly repair the tissue through fibrosis and scarring. They replace functional tissue with rigid collagen. And this is all controlled by chemical mediators like neurokinins.
Substance P, right. The text mentions substance P worsens the swelling and transmits pain signals.
Exactly. And inflammation is bad enough, but it becomes terrifying when it's aimed at the wrong target. Autoimmunity. Friendly fire.
Like Sjogren's syndrome.
Yes, in Sjogren's syndrome, CD4 T lymphocytes attack the salivary and lacrimal glands. So the patient has severely dry eyes and a severely dry mouth.
And then there's scleroderma, or progressive systemic sclerosis, which is massive collagen deposition and vascular disease. And there's that specific variant, CREST syndrome.
Yes, CREST is a great acronym for the boards. The C stands for calcinosis, painful calcium deposits.
And the R is Raynaud phenomenon, right, where the blood vessels in the fingers spasm from the cold and the fingers turn stark white.
Exactly. The E is esophageal dysfunction because the smooth muscle gets replaced by rigid collagen. The S is sclerodactyly, tight skin on the fingers.
And the T is telangiectasia, the dilated capillaries on the skin.
Spot on. And for diagnosis, you look at autoantibodies. CREST variant has anti-centromere antibodies, while the diffuse scleroderma has anti-CL70 antibodies.
Okay, so chronic inflammation and vascular damage inevitably ruins the body's plumbing. That brings us to systemic breakdowns, starting with edema.
Right. Edema is just fluid escaping into the tissue. And it's governed by Starling forces. Hydrostatic pressure pushes fluid out of the vessel.
Like in congestive heart failure. The heart isn't pumping well, so blood backs up, hydrostatic pressure spikes, and fluid is physically forced out into the legs or lungs.
Exactly. And opposing that is oncotic pressure. The blood has massive proteins, primarily albumin, which act like a chemical sponge, pulling fluid back into the vessel.
So if you lose that albumin, you get edema too, hypoproteinemia, like in nephrotic syndrome where the kidney leaks albumin into the urine.
Or cirrhosis. The liver is the factor that makes albumin. If the liver is scarred from disease or alcohol, it stops making it.
So the pulling force vanishes and fluid leaks out everywhere, whole body swelling. And if this hemodynamic failure gets worse, you hit the end stage, shock.
Shock is systemic hypoperfusion. Blood pressure drops so low that the organs just aren't getting oxygen.
Whether it's hypovolemic from bleeding out or septic shock from a massive infection, every organ suffers. The kidneys get acute tubular necrosis, right?
Yes, ATN. The tubule cells die and slough off. Clinically, urine output drops, and under the microscope we see dirty brown granular casts.
Basically, microscopic body bags of dead kidney cells.
Right. And the adrenal glands face a violent fate, especially in meningococcal sepsis. They suffer massive hemorrhagic necrosis.
Waterhouse-Friderichsen syndrome, they literally bleed into themselves and destroy their own tissue.
While the lungs develop hyaline membranes in the alveoli, making oxygen exchange impossible, and the heart gets tiny petechial hemorrhages.
It's a terrifying cascade, from one misfolded protein to multi-organ failure. So to fulfill our educational mission, let's test what you, the listener, have learned with a quick scenario.
Let's do it.
Imagine a patient with chronic acid reflux. The squamous cells in their lower esophagus change to simple columnar cells to survive the acid. What is this cellular adaptation called?
That reversible change is metaplasia.
Nailed it. Now, what if those new cells start growing in a disordered, irregular way with enlarged nuclei?
That dangerous pre-neoplastic growth is dysplasia.
Perfect. To wrap up, we want to leave you with a profound biological paradox from the text about aging. Biological aging is driven by the shortening of telomeres.
Right. Telomeres are the protective caps on the ends of our chromosomes. But every time a cell divides, the copying machinery can't quite reach the very end.
So they get shorter and shorter.
Exactly. And eventually they get so short the cell just stops dividing and undergoes senescence. It's a strict biological clock.
So here's the paradox. Scientists are looking for ways to artificially lengthen telomeres to stop aging. But if we figure out how to give our cells biological immortality, wouldn't we just be giving them the exact lifespan they need to bypass the proteasome bouncers, accumulate mutations, and become malignant tumors?
It's a razor-thin line between the fountain of youth and the origins of cancer, isn't it?
It really is. Something for you to think about the next time you consider the millions of microscopic battles happening inside you right now. Thanks for joining us on this deep dive.